Hello everyone. Am I audible?
Yes, ma'am.
Okay. Okay. Is my presentation visible?
Yes, ma'am.
Okay. So I think we shall begin. So welcome to NPTEL Experimental Biotechnology by Professor Vishal Trivedi from IIT Guwahati, and I'm Sumati, one of your TAs from University of Hyderabad. So this is live session for week 4, and we will be covering the topics for the assignment questions in week 4. So, if anytime I'm not audible or if my presentation is not visible, please tell me over the mic. Like I said before in the sessions, I won't be checking the chat box during the presentation. And if you have any questions, also in the middle, you can turn on your mics and let me know your doubts and queries. Okay. So let's begin with the first topic, that is Michael Sweat and the discovery of chromatography. Let me just turn on the pointer. Yeah. So Michael Sweat was born in 1972 and was there till 1919, and he was a Russian Italian botanist. So around 1901 to 1903, he was studying something called as plant pigments, which we know are of various colors, whether in the plants or like in the leaves or stem or in the flowers. So he needed a way to separate the chlorophyll from other leaf pigments. So the leaf is mostly green because of the chlorophyll, but it also has certain other pigments. So he was trying to study those pigments. So he wanted a way to separate these pigments from chlorophyll, which is abundant in the leaves. So he conducted an experiment. He prepared the extract from the green leaves of the plants, and obviously it had mostly chlorophyll, but along with that it has certain other pigments like carotenoids, xanthophyll, etc. So this extract was poured through a glass column packed with a solid absorbent, which could be calcium carbonate or sucrose powder. So you can see here in the picture, you have a column. This is a column which has a valve opening at the bottom. So it's basically like a tube. So it he filled it with a solid adsorbent which can absorb materials or molecules or chemicals. Here he had used calcium carbonate, and then as the extract moved down, so he added the extract into the column. Then he saw that different pigments bound to the adsorbent to different degrees. So the due to their adsorb difference in the adsorbing property on the calcium carbonate, different pigments separated like this when you add any organic solvent like ether. So you can see how from the plant extract which he had added over here, all these pigments started separating because each pigment had a different capacity to adsorb onto the calcium carbonate column. So Michael Sweat invented chromatography in 1900 during his research on plant pigments. He used liquid adsorption column chromatography with calcium carbonate as the adsorbent and petroleum ether or ethanol mixture as eluent. So, an eluent is a solution which you use to extract the things from the column. Like it will uh, dissolve the material which you need within the solvent, and as the solvent releases from the column, your desired chemical or pigment or anything will elute out along with the solvent. So here the solvent or eluent that was used was ether or petroleum ether or ethanol. Then he separated chlorophyll and carotenoids from the plant extract. So as the extract moved down, different pigments bound to the adsorbent to different degrees was seen. Then pigments separated into distinct colored bands: green, yellow, orange. So you can see here how different colors of pigments have separated into distinct bands. So this was the first visual demonstration of chromatography, which literally means color writing. So, calligraphy, right? Calligraphy is handwriting. So chromatography is color writing. Chrome or chroma means color, and graphy means writing. So this was the first ever visual demonstration and invention of chromatography. So, why did he use plant extract and not others? So, plant pigments are naturally colored molecules. So, the separation could be seen with naked eye. So, even if, like, it can separate different molecules or different substances, but unless they have color, you cannot visualize it with your own naked eyes. You might need some staining or something else later. But since the plant extract has colored molecules, since it was pigments, you don't need any other extra staining, and different colors of pigments had easily separated, which we can visualize with our eyes. Synthetic color mixes existed, but Sweat specifically wanted to solve a biological problem, isolating chlorophyll for photosynthesis research. And also, like, he was working on the plant biology. Since he was a botanist, he had some specific research going on with the plant extracts and chlorophyll. So he used plant extract to separate to separate them in chromatography. So, what is the significance of this? He showed that complex mixtures can be resolved into components based on their interaction with a stationary phase. Excuse me. So, here the stationary phase is calcium carbonate, which he had added into the column here. So, this becomes your stationary phase because this does not come out of the column, it will remain in the column. And whatever extract and ether are all mobile phase. So uh, he resolved these components. Resolved means separation into distinct bands like this. So he resolved these components based on their interaction with the stationary phase. Like I said, the different components have different affinity to the solid phase, that is the adsorbent. It has different capacity to adsorb with the calcium carbonate. So based on their interaction, he had resolved it into distinct bands, and a mobile phase which is a solvent. So here what is the mobile phase? It's the ether. So ether moves along the solid phase, the stationary phase, carrying out or eluting out your components. So your stationary phase is the adsorbent, which is calcium carbonate, and your mobile phase is the solvent, which is either petroleum ether or ethanol. So this principle became the foundation of modern chromatography techniques. So since like this was the first ever experimental chromatography, this was later again used into and modified into different types of chromatography depending upon the type of separation, the particle, the nature of the particle which you want to separate, etc. So, since then, how chromatography has evolved is in the 1940s, Martin and Sinch started with paper chromatography. Then in the 1950s, we saw thin layer chromatography or TLC. And then we also saw gas chromatography during that time. And during the 60s and 70s came the high performance liquid chromatography, which is still used very extensively in separation techniques. Also, gas chromatography is highly used. So, this uh, Sweat's chromatography was a foundation for development of all these kind of chromatographies. So today we have many advanced forms of chromatography like affinity chromatography, ion exchange chromatography, and 2D chromatography and LCMS coupling chromatography, etc. So it gave rise to many different types of chromatography techniques. So next is the distribution by surface area. So in chromatography method, distribution by surface area, we see in case of hydrophobic interaction chromatography. So why in hydrophobic interaction chromatography? Because the proteins or the molecules in this have hydrophobic regions. So, hydrophobic means that they repel water or they do not like water. Hydro means water, phobic literally means scared. So, they are scared of water. The so they try to stay away from water molecules as much as possible. And then there is something opposite to that, which are more like which need water and which likes the water environment, is called as hydrophilic region. So proteins and biomolecules often have certain regions which are hydrophobic in nature, that is, they do not want water molecules surrounding those regions. So they will attain a particular conformation to avoid interactions with water in those particular areas. So these hydrophobic regions might also be present on their surfaces. So the extent of hydrophobic surface area exposed varies amongst the proteins. So different proteins have different surface area of hydrophobic regions exposed. Like, a protein is not like a chain or a 2D structure. Protein is a 3D structure and it has like conformation in which it folds. So, depending upon the folding and the conformation, different proteins have different uh, amount of surface area of hydrophobic region which are exposed. So, this depends on the folding, modification, etc. etc. So, in HIC, that is in hydrophobic interaction chromatography, the stationary phase is coated with a hydrophobic liquid. So, hydrophobic liquid is also similar to the hydrophobic region. So something like butyl or octyl or phenyl groups. So when a salt rich buffer, that is a buffer in which you have like high amount of salt is used, like for example ammonium sulfate, the salt removes the water molecules from around the proteins, and then the hydrophobic surfaces become more sticky, so it helps your hydrophobic surface to bind. Then proteins with more hydrophobic surface area bind more strongly to the column. So your salt basically helps in enhancing the binding capacity of your hydrophobic surface area of your proteins. Then during the illusion, gradually you start to lower your salt concentration. So we had initially used a buffer which was rich in salt or which was high content salt. Now this helped the protein to bind to the column. Now once it has bound, you need your protein to elute out. You need your protein to come out of the column. So then what you will do, you will start gradually lowering your salt concentration. So what will this do? This will start uh, the loss of the binding of your protein from the column. So the proteins are then released in order to least hydrophobic to most hydrophobic. So the least hydrophobic will have the least interaction in salt rich buffer. So when you start reducing the salt content in the buffer, your your protein which has least hydrophobicity will now elute out faster. So you can see here this is a setup, or this is a chromatographic support. These are your hydrophobic ligands attached to it using via a spacer arm. And this is your protein, and these are the regions of your hydrophobic surface, hydrophobic zones or hydrophobic pockets. So when you have salt-rich buffer, your protein will be now attached to your uh, hydrophobic ligand or your resin. And once you start reducing the salt content, your proteins will start eluting out in the order of the amount of hydrophobic region it has. So this was how the distribution of surface area is important in case of hydrophobic interaction chromatography. So in case of affinity chromatography, it depends on specific binding pairs. So, as the name suggests, affinity chromatography means interactions or particular pair of proteins or pair of interacting molecules. Like for example, your antibody and antigen complex, your enzyme substrate complex, their affinity is very specific to that particular antibody to antigen and that particular enzyme to that particular substrate. So this is what comes in use in case of affinity chromatography, and this is independent of any type of hydrophobic surface area. That means the hydrophobicity of the area does not affect your affinity chromatography. Then, in case of ion exchange chromatography, separation depends on the net charge of the protein at a given pH. So, ion exchange, again, as the name suggests, it's ionic in nature, so it is dependent on the net charge of the protein or the ionic charge of the protein at a particular pH, because at different pH, different proteins have different net charge. So the surface area also does not determine the retention here. It is only the charge density that comes into play in case of ion exchange chromatography. And in case of gel filtration chromatography, it's also called as size exclusion chromatography because it depends on the size and the pore size of the column. It is based on the molecular size and shape of your biomolecule or your protein of interest. The larger molecules elute from the pores and elute earlier, and the smaller ones enter the pores and are delayed. So here, when you have your beads or your resins here, they have pores, and then between your beads or resin, you have gaps. So if your molecule is larger, it will not enter the pores of your beads, and it will escape in the gaps between the beads or the resin, and it will run faster. And your smaller molecules will enter into the pores and will take time to move down. Then here also surface hydrophobicity does not play any role. It's only the molecular size and the shape. Then hydrophobic interaction chromatography separation is directly related to the extent of exposed hydrophobic surface area. So larger hydrophobic patches, stronger will be the retention in the column. Like we said, when you start eluting it with lower salt concentration buffers, your first molecule that will exit will be the one which has the least hydrophobic patch or the least amount of hydrophobic region that will be exposed in the surface area. So, what are the applications of HIC? It is used in purification of monoclonal antibodies and recombinant proteins. It can be used in separation of protein isoforms, which has difference in their hydrophobicity, and to study protein folding or unfolding, which changes the exposure of hydrophobic regions in the protein, and useful in downstream processing where gentle, non denaturing conditions are needed, unlike reverse phase HPLC, which uses organic solvents. Then HIC analytes are separated by amount of hydrophobic surface area they expose. unlike charge, size, or specific binding, which is seen in case of ion exchange, gel filtration, and binding affinity chromatography. So the next one is distribution coefficient or partition coefficient, which is KD, is given by concentration in phase A and concentration in phase B. So distribution coefficient or partition coefficient is the equilibrium ratio of a solute's concentration in two immiscible phases. So you have like an aqueous phase and an organic phase. So these phases are immiscible. Immiscible is the liquids do not mix with each other, so they are always in a phase separated manner. So when you add certain solutes in such kind of phase separated immiscible liquids which are together, you see that the solutes will dissolve in each solution in different concentration. So some solutes might dissolve more in your organic solvent and less in your aqueous solvent, and some other solute or the solutes of some other chemical might dissolve more in your aqueous solvent than in your organic solvent. So that is why it is defined as how your given solute gets distributed in the phases of two immiscible liquids or solvents. So, generally, partitioning is the movement of a drug molecule or any biomolecule from one phase to another phase or vice versa. So, once they are in the solution or in the phase separated liquid phases, they will maintain an equilibrium. Then there might that is maintained by movement of the molecules from this phase to this phase or this phase to this phase. This is called as partitioning, and this is your movement. And the constant which helps you define this for every solute or every or each chemical is called as distribution coefficient. So if A is organic or stationary phase and B is aqueous or a mobile phase, then the distribution coefficient is given by the concentration of the solute in A by the concentration of solute in B. So this is B. Second. Yeah. So the distribution coefficient is given by the concentration of the solute in liquid A and divided by the concentration of the solute in liquid B. It's a constant, and at a given temperature for a fixed chemical, like I said, for a particular chemical, the number or the ratio is constant at a given temperature. So pH ionization affects the distribution coefficient of any chemical. Unless these variables remain constant, your KD value will be same for a particular chemical. So in this case, your ZA is nothing but the concentration of your solute Z in phase A or solvent A, and concentration of ZB is concentration of solute in phase B or solvent B. What exactly is distribution coefficient? So, in simple terms, imagine you have a solute or a molecule that is placed between two immiscible phases which do not mix. So you can see there is a clear separation between your octanol here and your water. Water is your aqueous phase, octanol is your organic phase. So the organic doesn't mix with the water doesn't mix with the organic. So there is clear separation and they are immiscible. So your solute Z, just assume this is your solute Z. It is placed between these two immiscible phases like water and octanol. At equilibrium, you can see there is movement, and once the movement is done based on the distribution coefficient, they will attain equilibrium. So at equilibrium, some molecules of Z will dissolve in phase A and some molecules in phase B. You can see this, the the solubility is more efficient in organic phase for this than in water. So more number of solutes have been distributed in the organic phase, and very less number of solutes have distributed in the water phase. So the ratio tells us the preference of the compound for one phase over the other. So in this case, you can see this solute prefers organic solvent more than aqueous solvent. So this ratio helps us determine the preference of a compound for one phase over the other. If we add plus, or or I- if you have added, like if you have done addition of your concentrations, or if you have subtracted your concentrations, it would not give a meaningful information about your equilibrium, because when you add or subtract your concentration, you are reducing or increasing your concentration of the solute, but it does not help us understand its preference in either of the solvents. And if you multiply, this would not describe the equilibrium phase. If you multiply, it will give you some exponential value, which will not help us understand its distribution in it. So we have to take a ratio, like how much is in one phase and how much is in another phase. Like for example, it is 5 by 10, 5 molecules in one phase, 10 molecules in another phase. So ratio here makes sense to explain its its distribution preference, or its partition preference. So there are different cases. Like for example, if your distribution coefficient is greater than 1, your compound prefers A. Like the example I gave was 5 by 10. So 5 by 10 is nothing but 1 by 2, which is 0.5. So this is not greater than 1. So this solution does not prefer phase A. For if your distribution coefficient would have been 10 by 5, then it is 2. So 2 is greater than 1, so that means it prefers phase A, because more molecules, 10 molecules have been dissolved in phase A, so obviously it prefers phase A. In the first case, when it was 5 by 10, the ratio was zero point 0.5. So that means it is less than 1 and it prefers phase B, because 5 molecules were distributed in solution A and 10 molecules were distributed in solution B. So if your coefficient is less than 1, it means that the compound prefers phase A more than phase B. So if your compound is equal to 1, if the Kd of your compound is equal to 1, that means it distributes equally between both the phases. Then coming to the applications, we have liquid extraction. You use this and for separating organic molecules from aqueous mixture. You can use this for drug designing, because partition coefficient is based on the principle of octanol by water system to check the drug hydrophobicity, and then you use it in chromatography, which helps you to determine the retention time of solutes, and in environmental chemistry you can use it to predict pollutant distribution in soil to water or in air to water, etc. So in these cases and many other different cases where you have two different phases and you need to determine the preference of a solute in one to the other, you definitely have your distribution coefficient being used in such scenarios. Then again, coming to hydrophobic interaction chromatography. So like we discussed, the basic principle behind the hydrophobic interaction chromatography is that proteins bind to the hydrophobic ligands, like your butyl, octyl, phenyl, etc., on a solid matrix in the presence of high salt content, that is your ammonium sulfate. So your high salt reduces the protein solubility, which forces the hydrophobic patches of your proteins to interact with the columns ligands, which are hydrophobic ligands. So proteins are then eluted by decreasing the salt concentration. We discussed this. So low salt restores the solubility. Once they have increasing solubility, they start eluting out. So during the operation, we work in high salt buffers, but that's different from storage. So you should not get confused between your working buffers and your storage buffers. Your high salt buffers are used when you start your experiment, when you put in your molecule in the column and you want now the molecules to bind to the column, you use high salt buffer, but you do not store it in high salt buffer. First of all, why columns need storage solution? Because after use, chromatographic columns are susceptible to microbial growth like bacteria, fungi, yeast. Microorganisms are ubiquitous in nature. So obviously they will also start growing in your columns because they have been moist, they have been used, etc., etc. So your microbial growth will be seen. So you need a storage solution to prevent from contamination from microbes. So the column contains sugars and organic ligands. So this is where your microbes thrive. They need sugars, they need organic ligands for their growth and survival. So it is a very nice attractant. It's a very nice environment for your microbes to grow and thrive. So you need to avoid this. So microbes thrive if stored in water, definitely. And then other than this, you also have ligand degradation. So your hydrophobic groups may get hydrolyzed and then it may degrade. So you want to prevent this because if your ligand gets degraded, you cannot expect your protein or your biomolecule to bind to the ligand because ligand is where your hydrophobic patch comes and binds. So if there is no ligand left in your column anymore, your molecule will just run out as soon as you add in the column. It will not bind. Then column blockage might happen. So precipitated salts, if stored in high salt buffer, it will precipitate over time and you will have salt crystals in your column, and once you have salt crystals in your column, it will block your column and you cannot run the column, you cannot elute anything out. So these are the main reasons why you need to have a proper storage solution for your columns after use. So that is to prevent microbial contamination, preserve the ligand integrity, and avoid salt crystallization or drying. So how do you do this? You use a solution of alcohol and sodium acid. So alcohol, that is mostly 20% ethanol or isopropanol is used because they help in uh, preventing microbial growth, and also if there is any microbial contamination, they help in degrading it. So it denatures the microbial enzymes and membranes and prevents microbial contamination, also keeps the column matrix hydrated. So whatever you're using is a certain percentage of alcohol. So it also has water content in it. So it will help your columns stay hydrated. Otherwise, the matrix might get dried up. Then we use sodium azide around 0.02 to 0.05%. This is also a potent antimicrobial agent and it is bacteriostatic and fungistatic. Works in very low concentrations, and the combination of ethanol and sodium azide is very useful for long term safe storage of your columns. This is the reason why we use alcohol and sodium azide. So, like we said, if you use high salt concentrated buffers, you will have salt precipitation in the buffers over time, and salt can crystallize during storage and clog the column. So salt solutions are generally not reliable, and also they do not prevent the microbial growth. Ammonium sulfate is used during HIC operation and not during storage. It can also precipitate and damage the matrix if stored for several days, weeks, or months. Then distilled water. Obviously, we discussed distilled water promotes microbial growth, both fungus and bacteria, and it also leads to growth of biofilm formation, and the column will like start smelling and it becomes foul, etc. Basically, it becomes unusable. So all these three will not help in any way if you store your column in that. The only solution that is useful here is your alcohol and sodium azide cocktail. Just a minute. Okay, just connect my charger. So okay, I hope my screen is still visible. Yeah. Yeah, so this was about storing the columns. Next is DEAE, that is Cefidex A50. So what is DEAE Cefidex A50? It is a DEAE is diethyl amino ethyl group. It is a weak basic group that carries a positive charge at neutral pH. And Cefidex is basically a polymer, so it's a dextrin bead and these are cross-linked dextrin beads used as the support matrix. So it is a porous polymer, so it helps us to run our chromatography in it. And A50 is a grade designation basically. The 50 indicates the bead size, porosity, and intended flow properties. So this is a material which is widely used as a stationary phase in ion exchange chromatography. How does it work? Because it has DEAE, that is diethyl amino ethyl group, which have positive charged at neutral pH. They help to bind with the negatively charged molecules like DNA, RNA, acidic proteins, and phosphorylated molecules. Basically, it's used as a positive matrix. So how the process works is first you have sample application. So your sample should be negatively charged for obvious reasons because your matrix or your stationary phase is positively charged. So the negatively charged molecules in the sample bind electrostatically to the positively charged resin. Then you have your washing steps. So here unbound molecules are washed away with the buffer. Then you can elute only the bound molecules. So next step is elution. So here your bound molecules are released by gradually increasing the salt concentration, like using NaCl or KCl, or you can also change your pH because the positive charge is at neutral pH. So when you change the pH, the positive start, if your positive charge becomes negative, then slowly all your negatively charged molecules will start eluting. So the salt ions compete with the bound biomolecules. So how your salt helps us? They compete with your bound biomolecules by displacing them from the resin. So they push away your biomolecule and sit on the resin. Then your biomolecule will be eluted out. So this type of chromatography is seen in case of ion exchange where DEAE is used. for anion exchange, and for cation exchange we use CM Cefidex, which has carboxymethyl groups. So carboxymethyl groups will have negative charge on them, so they help in binding the positive ions, that is your cations. So this principle fits for ion exchange chromatography, anion exchange chromatography. We have DEAE, resin is positively charged and binds to anions which are negatively charged. Cation exchange chromatography is CM Cefidex with carboxymethyl groups. The resin is negatively charged, so it binds to cations which are positively charged. The others, like in case of gel filtration, it helps in separation by size and shape of the molecule. The stationary phase is porous beads like your Cefidex G75 and G200, and DEAE Cefidex is not porous size exclusion gel. It has charged. So DEAE is used for ion exchange and not in gel filtration. And hydrophobic interaction chromatography, it is a separation based on the hydrophobicity. The stationary phase is ligands, hydrophobic ligands like butyl or octyl phenyl. Here also DEAE does not uh, have any use because DEAE is hydrophilic in nature, so therefore it cannot be used in hydrophobic chromatography. Then affinity chromatography, which separates molecules based on the specific ligand binding, like the antigen antibody or histag nickel NTA, nickel NTA, and protein A and IgG binding. These are specific ligand binding partners, and DEAE does not have any affinity. It is only charged base separation. So DEAE is not used in any of the three here. It is only used in case of ion exchange, that too in case of anion exchange chromatography. Next is the effect of salt on protein solubility. So proteins in aqueous solution, they have hydrophilic and hydrophobic regions. So in water, these hydrophilic groups will interact and the hydrophobic regions will be usually closed, or it will be hidden away from being exposed to water. So proteins have hydrophilic, that is charged or polar, and hydrophobic, which are non-polar regions. So in water, that is in aqueous solution, hydrophobic regions interact with the water molecules, so they are basically exposed to the water molecules, while the hydrophobic regions are usually buried inside. So like we spoke about the protein folding, which also is dependent about the hydrophobic regions in the protein. The protein will usually fold in a way in an aqueous solution, if it is in an aqueous solution, in a way that the hydrophobic regions are usually buried inside the core of it and it will not be exposed. The exposed regions will be your hydrophilic regions. So the electrostatic repulsion between similarly charged proteins can sometimes make the proteins unstable in solution. Sometimes proteins might have interactions with each other, and if there are any sorts of electrostatic repulsion between the proteins, then this sometimes affects its unstability in the solution. So then what we do, we have something called as salting in and salting out. Initially we add salt to it, which is called as salting in. So when you add a low concentration of salt, salt ions will shield the charges on the protein surface, so the electrostatic repulsion between the proteins decrease. The proteins will then interact better with the water molecule, therefore the solubility will be more. So one example of this is adding of NaCl or ammonium sulfate at low concentrations, which often helps in dissolving or solubilizing the proteins more efficiently. Then in case of high salt concentration, which is salting out, at high salt concentrations, sorry, at high salt concentrations, salt ions compete with the water molecules. So when you keep on adding salt in it, now salt is more. So therefore salt needs the water for it. Then it will not let your protein interact with the water. Only all the salts only will start interacting with the water. So then proteins lose their hydration shell, and then hydrophobic patches become exposed. Like I said, the hydrophobic core is present inside, and then you have a shell or a protecting outer hydrophilic, uh, like a protective layer. So when that hydrophilic region is not able to interact with the water, it will obviously change its conformation, because now it doesn't have to be in outside, it can be anywhere because it has lost interactions with water. So then this will lead to the inner core of hydrophobic patches to become exposed now. So then this leads to aggregation and precipitation. Basis of ammonium sulfate precipitation, a common protein purification step. So you usually see, like, if you have some kind of proteins that have been dissolved, or anything, any chemical that have been dissolved, maybe due to wrong storage or due to addition of more salt or wrong conditions, they will precipitate out. Or if you want to extract something also, you add some kind of salt buffer or something to precipitate them. So this happens because the protein loses its hydrophobic shell, hydrophilic shell, and your hydrophobic patches become exposed, and it loses interaction with the water, and it gets aggregated, and it gets precipitated. So this is seen in case of salting out. So here you can see your solution, solutes in your solution, and your salt is added to it, and then you see precipitation here. So this is your salting out effect. And then when you have low salt concentration, you see a homogeneous solution, which is called as salting in, where there is good solubility or increase in solubility of your molecule. Then why this happens is because low salt shields the charges and increases the solubility in simple terms, and high salt reduces the water availability, so this will decrease the solubility. This dual effect makes a bell-shaped curve if you plot the protein solubility versus salt concentration. So basically, if when you see with low salt concentration, the protein solubility is high, and then when you increase the salt concentration, the protein solubility goes to a certain extent, it keeps increasing, and once it reaches the threshold of it, and then the solubility starts dropping. So you have your normal distribution sort of bell shaped curve, because in the initially, if your solubility, imagine this is an x-axis, and if you have your y-axis as salt concentration, and your x-axis is your solubility. So initially the solubility is low, so then you start adding salt. So once you start adding salt, the solubility will start to increase. But at one particular point, uh, when the salt concentration is too high, and if you keep on increasing your salt concentration, then your solubility will decrease. So it will give you a bell-shaped curve. Then basically, initial salt helps proteins to dissolve better, but too much salt pushes them out of the solution. So you need to have a standardized salt concentration or an optimal salt concentration for proper dissolution or properly solubilizing your proteins. So this is just a comparison. Low salt, salting in; high salt, salting out. Salting in increases solubility, salting out decreases the solubility and basically precipitates out. So basically salting in is pushing your protein to dissolve inside the solution more. Salting out is pushing your protein out of the solution to precipitate. So in case of salting in, salt ions shield the electrostatic charges and prevent the aggregation, that is more solubilization. In case of high salt, salt competes with water, exposes the hydrophobic patches, and pushes your protein out, leading to its aggregation or precipitation. Hope this was clear. Then Next, again, we have HIC and salt equilibration. So, like we discussed, it's the why high salt buffer is needed. That is, in aqueous solutions, proteins are naturally surrounded by a hydration shell of water molecules bound to their polar groups. Hydrophobic patches on the protein are usually hidden or buried inside where water molecules repel them. So, when high concentration of salt is used, like your ammonium sulfate or sodium chloride or sodium citrate, salt ions compete with the proteins for water molecules, and hydration shell around the protein gets disrupted. Hydrophobic regions are exposed. This promotes the hydrophobic interactions between the protein patches and the hydrophobic ligands in HIC stationary phase, that is your butyl phenyl octyl groups bound to the agarose beads. So basically, it will push away your protein molecules. Then it helps, uh, however, it will help in the hydrophobic interaction between your protein patches, because now your protein patches are exposed. So here what we saw is high concentration is exposing your hydrophobic uh, regions of your protein, and it is leading to aggregation. If you make it as a use, if you take advantage of this, you can use this method to expose your hydrophobic regions when you want them to be interacting with something in your chromatography. So this will help in your hydrophobic interactions between your protein patches, which are now exposed, to be available for your hydrophobic ligands in your stationary phase. So this is how you can use your salting out to increase binding in your HIC resin. Then equilibration step. So before loading the proteins, the column is equilibrated with a buffer containing high salt, usually 1 to 2 molar ammonium sulfate in phosphate or tris buffer, because tris buffer or phosphate buffer helps in maintaining the pH. So this has a neutral pH of 7 now. Then the hydrophobic ligands on the stationary phase are surrounded by a salted out environment. Then when the protein solution is applied to it, the protein immediately binds strongly via their hydrophobic patches, because now the patches are exposed, the hydrophobic ligand is also exposed, and it will easily bind as soon as you add it. Now, protein binding and separation. So, different proteins have different degrees of hydrophobicity. So, strongly hydrophobic proteins bind very tightly to the column, and less hydrophobic proteins bind weakly, or maybe they may not even bind to the column if they have like very less hydrophobic region in their structure. So now when you elute, your least bound will elute out first, because you start decreasing the salt concentration in the mobile phase. So you have a gradient of salt, like you have initially, what was it? It was 1 to 2 molar. So now you will decrease the buffer. Slowly you will use a zero point 0.8 buffer, then you'll use a zero point 0.5 buffer, then you'll use a zero point 0.1 buffer. So in a gradient of buffer, you start eluting your proteins. So as the salt decreases, the hydration shell is restored, and hydrophobic interactions weaken. So your proteins elute in order of increasing hydrophobicity, that is, least hydrophobic proteins come out first. In other cases, if you use water or methanol or acetonitrile, what happens is water would maintain full hydration shell around the proteins, and then there will be no interaction of your hydrophobic region of your protein with your resin. So no significant hydrophobic interaction occurs. The protein won't bind to the column at all in the first place. If you use an organic solvent like methanol, it is used to reverse the phase. It is used in reverse phase chromatography and not in hydrophobic interaction chromatography. And also, methanol denatures the proteins at high levels. Then acetonitrile. It is also an inorganic solvent, and it is typically used in reverse phase chromatography or reverse phase HPLC in most cases. And this is also not suitable for your hydrophobic interactions, because it disrupts the native structure of the protein. So what you use here is your high salt concentration. Then coming to cation exchange chromatography, the basis of separation here is your charge of the molecule. It specifically is positively charged molecules, that is your cations. They bind to the stationary phase while the neutral or negatively charged molecules will pass through. Here, the stationary phase is modified to have a negatively charged functional group so that it can bind to your positive cation. Then common examples of your stationary phase is like the CM, that is your CM Cephidix, which has the carboxymethyl groups, which is a weak cation exchanger. Or you can also use sulfonic acid, which is also a negative charge, and it is a very strong cationic exchanger. Then coming to the role of pH and proteins isoelectric point here, proteins that carry charge depending on pH, that is at low pH, that is if the pH is lower than the pi, that is the isoelectric point, then the protein is positively charged and it binds to the uh, cationic exchanger. If your pH of your protein is more than if your pH is more than the isoelectric point of your protein, then the protein is negatively charged and it will not bind. So since this is a cation exchanger, only positively charged protein molecules can be bound and can be exchanged in the chromatography. If your protein's pI is more than the pH that less than the pH that you're using, it will have a net negative charge. So a negatively charged protein will not bind to a cationic exchanger. This makes the pH selection very critical to decide which proteins bind. Next is the process steps. So first is column equilibration. You have to equilibrate the column. That is, the resin is equilibrated with low salt buffer first at the chosen pH depending on the isoelectric point of the protein you want to use in the chromatography. Then you load your sample. So your positively charged proteins bind to the resin. Neutral and negatively charged proteins flow through completely. Then you do washes. This is to remove excess or remaining neutral charged molecules or negative charged molecules if anything is remaining. And also, if something is bound very weakly, like it has very less positive charge on it, even those kind of impurities will be removed in your washing steps. Finally, you will elute out all your positively charged proteins. Now you initially we had used low salt buffer which helped in the binding. Now you will use high salt buffer to elute out. So you increase the salt concentration like the sodium chloride, in which the sodium will now become your cationic exchanger and it will compete or exchange your positive proteins which is bound on the resin. So salt cations compete with the bound proteins for resin binding sites. So they push out your positively charged protein and they go and take their place. So your positively charged protein will now elute out in your eluent. So weakly bound proteins will elute out first, obviously, and the strongly bound or tightly bound proteins will elute out at the end. Alternatively, you can also change the pH because, like we discussed before, the pH is very important for binding. So, once bound, if you change the pH, depending on whether it is greater or lesser, your protein can start eluting out in your buffer because the binding will decrease. So, protein pH will a change in pH will alter the protein charge and release it from the resin. What are the applications of this? So this can be used in purification of proteins like your positively charged proteins from a mixture of neutral, negative, positive protein and peptides and nucleotides, etc. Separation of isoforms of the same protein with slightly different charges. And then industrial scale protein purification also is done for monoclonal antibodies, etc. So therefore, your cation exchange chromatography relies highly on electrostatic attraction between the positively charged biomolecules, that is your cations, and the negatively charged resin groups. By carefully adjusting the pH and the salt gradient, the proteins can be selectively bound and eluted out. So this is a table which you can compare your cation exchange, anion exchange, gel filter, HIC, and affinity chromatography. So in cation exchange, it is based on positive charge proteins. In anion exchange, you have your negatively charged proteins binding. In gel filtration, it is size and shape dependent. In HIC, it is hydrophobicity that determines the binding, and in affinity, it is specific binding of ligand receptor pairs. So the stationary phase used in all these different chromatography. Coming to cation exchange chromatography, we use CM Cephros or SP Cephros. In case of anionic exchange, we use DEAE Cephros or Q Cephros. And then for gel filtration, we use Cephedex G series. And for HIC, we use phenyl Cephros. And for affinity chromatography, we use the specific binding pairs like nickel NTA and protein A IgG pairs. So these are your examples of stationary phase for different types of chromatography techniques. So before going to the next topic, uh, does anybody have any question or any doubt? We can probably take a 2 minutes break. If you guys have any questions in the meanwhile, you can type it in the chat box or you can ask me. So it's 4:13 right now. We will resume back at 4:15. Okay. So in the meanwhile, you can check out the questions if you have any doubts, or if you have any doubts in whatever I have explained till now, you can go ahead and write it in the chat box. I'll be back in 2 minutes. Yeah, okay. So I guess there are no questions, so we can resume. I hope I'm audible. Let me know if I'm not. So the next topic is the chronological order of movement of mobile phase in a chromatography system. I hope my slides are still visible. Okay. So first, you have your solvent reservoir, which is a mobile phase storage system. So this is the starting point of it, and it contains the mobile phase, which can be your water or buffer or the solvent which you're using, depending upon the type of chromatography system or chromatography you're using. And in ne- gradient chromatography, multiple reservoirs may be present, which might have different solvents in them. If you're like having multiple solvents initially, like your water or your acetonitrile, etc., then comes the pump. So the pump pulls the mobile phase from the reservoir and pushes it forward with controlled flow and pressure. So you have all the parameters, you can adjust it with particular flow rate and pressure with which you push your mobile phase into the column. This ensures reproducibility of retention times, and high performance systems like HPLC and FPLC use high pressure pumps to drive the liquid through tightly packed columns. Then comes sample injection loop. So here in the sample injection loop, we inject the sample. So sample is introduced here into a continuous stream of mobile phase, and in the manual systems, we may use a syringe to inject the sample, or you can also have sample automated samplers which will automatically inject your sample when you have loaded it. So in automated systems, it is done by an auto sampler, and we have injection loops which are usually 20 to 100 microliters capacity and load a fixed sample volume without disturbing the flow. So next comes your column, in which all these are now flowing. That is where the separation happens, and the stationary phase is present in your column, which could be anything depending upon your experiment and your protein. It can be silica, it can be resin, it can be beads, anything you are using, particularly in your using your different tag or different type of techniques that you're using. So this interacts differently with each molecule. So based on the technique, if it's ion exchange, you have charge, you in gel filtration, you have size, in affinity you have specific ligand binding, in reverse phase you use hydrophobicity. Then after the column you have a detector. So now once it has moved into your stationary phase, now you have to elute it. So as molecules elute, they pass through a detector. Once it has eluted, you have to detect what is being passed through. So you have common detectors like UV absorbance for proteins and nucleic acids, fluorescence for sensitive biomolecules, conductivity for salts and ions, and refractive index for general molecules, sugars, and polymers. The detectors will send signals to the data system to generate chromatograms. So in case of UV it will take absorbance as the proteins or nucleic acids pass, and your absorbance will be plotted, and fluorescence also same, conductivity also same, refractive index also same. So then the data system will use these numbers to plot a graph, and depending on the proteins and or biomolecules eluted, you will get a peak. Then after that you have a fraction collector. So now it has eluted, you have to collect your samples after separation. So you have something called as a fraction collector. So it collects the separated components into tubes or wells for further study. You can also have, you can collect it manually if you're using a manual column, or if it's an automated system, it will have an automated fraction collector. It will automatically collect different samples in different tubes. So this is often synchronized with the detector, because as soon as it gets one peak of a component, it will collect it in one tube. Then if there is a different component, you will get a different peak, it will collect it in a different tube. So this will maintain the purity of the fractions, and it is useful in preparatory chromatography, where you do protein purification, or if you have to isolate any specific compounds. So you have your pure fractions collected at different tubes. So based on your column, based on the illusion fraction where you have received the peak of your particular protein of interest, you can collect that particular tube number. You can see the volume, like if your protein of interest has eluted at say 500 microliters, then you will collect the tube in after 500 microliters have eluted. If like five tube, if 100 microliters have eluted and you have your fifth tube, so your 500 microliters will fall in your fifth tube. So in that way, with respect to your peaks and illusion fraction, you can take the appropriate tubes in which you will have your pure fraction of protein. Then coming to butyl S suffrose. So hydrophobic interaction chromatography works by separating proteins based on hydrophobic patches on their surface, which we discussed. And the stationary phase in HIC is made of agarose or sapphirose beads that are chemically modified with hydrophobic groups like butyl octyl phenyl. So we said the stationary phase in HIC is butyl octyl phenyl. So these groups are attached to the agarose or sapphirose beads. The proteins that bind to these hydrophobic ligands in high salt content, due to the enhanced hydrophobic interaction, they are often eluted gradually by reducing the salt concentration, which decreases the hydrophobic interaction and releases the protein. So why butyl S suffrose is the right stationary phase in HIC? So in HIC, since we know proteins bind by the surface hydrophobicity under high salt conditions, the stationary phase is designed in a way to inert support to the covalently coupled hydrophobic ligands. So you have your sapphirose or agarose which are coupled to your hydrophobic ligands. So butyl S suffrose beads with butyl, that is C4 ligands, have the classic HIC resin. So the proteins with more exposed hydrophobic patches bind more strongly. Other options like Carbopac MA1 is a anion exchanger. So for carbohydrates, and it cannot be used in HIC. And your Dynex Carbopac PA1 is also an anionic exchanger for oligosaccharides. And your Q suffrose is a strong anionic exchanger, quaternary amine used for ion exchange and not in HIC. So for in HIC, you will use butyl as suffrose. So how it works is you equilibrate your column. Here also you have your high salt buffer with which you equilibrate first, then you load your sample. Because of the high salt buffered condition, your hydrophobic sample will bind easily to your resin. Then you elute by decreasing your salt concentration. Then you break the hydration, you bring back the hydration shell, and your hydrophobic interactions are now weakened, and your proteins start elusing in increasing order of hydrophobicity. Then you regenerate and clean the column. How do you do is you use low salt concentration first, and then a mild organic or detergent wash if needed, and then store in a preservative, which we discussed was 20% ethanol in sodium azide. So for choosing HIC ligand strength, butyl, that is C4, is milder hydrophobicity and gentler on the sensitive proteins, and it's a good starting point. If you want a higher hydrophobic ligand more than butyl, you can go for octyl, which is C8, which has intermediate strength. If you need much stronger interactions, you can go with phenyl, which has very strong interactions, and it is useful for very hydrophobic targets or when binding with butyl is weak. So always starting point, you go with butyl because your proteins might sometimes be sensitive, and with higher ligands it might denature. So you go with butyl. If there is no binding, you can increase it. Go with octyl. Still no binding. You need more stronger interaction. You can go with phenyl. So again, coming to salting out. Salting out is a protein precipitation method caused by very high concentration of salt, usually ammonium sulfate. So when too much salt is added, proteins lose solubility and aggregate, eventually precipitate out of the solution. So the mechanism is normal solubility in low salt. Proteins have charged groups on their surfaces. You have your carboxyl group, you have your amine group. Water molecules form a hydration shell around the proteins, so keeping them dissolved. In addition of moderate salt, which is salting in, your salt ions shield the protein charges. So they help in maintaining the charge, and electrostatic repulsion decreases between other proteins. So proteins stay dissolved and stable. But when you increase the salt, addition of high salt leads to salting out, and the salt ions strongly attract water molecules now. So they compete with the water molecules, they compete with your proteins for the water molecules. So less water is available for your solvate proteins. So hydrophobic patches of the proteins now come together. Protein protein interactions occur and they aggregate and they precipitate, and your proteins precipitate in the solution. How can you use this technique? Like we discussed, we can use this to precipitate your particular proteins, to separate proteins based on their solubility. Each protein has a different salting out point, like your isoelectric point, which differs from protein to protein at different pH. You also have different salt concentrations for a particular protein at which it precipitates. So this allows partial purification. Industrial uses are enzyme extraction for proteases, lipases, antibody purification from serum, then sample preparation for chromatography. Proteins can be salted out, collected, and rediscovered for further purification. So basically, salting in is more soluble, salting out is less soluble, and precipitation. This is how much you have to remember. Then again, coming to butylate sulfurose column. So, like we discussed, butylate sulfurose, it is a type of hydrophobic interaction chromatography resin. The base matrix is cross-linked agarose beads or sulfurose beads, and these beads are chemically modified with butyl groups, that is your C4 group. Then butyl groups are moderately hydrophobic. They interact with hydrophobic patches in the protein. This is ideal for separating proteins partially hydrophobic but not bound too strongly. The main principle of interaction. In the presence of high salt concentration, water molecules are pulled away from the protein. Hydrophobic patches on the protein become exposed. These hydrophobic patches interact with the butyl on the resin. On lowering the salt concentration, interactions weaken and proteins elute. So since we discussed this, I'm going a little faster. So this is a comparison between the different sulfurose beads with methyl butyl octyl groups. So methyl is the weakest because it is single carbon and hydrophobic strength is very less, and it is typically used for very mild separation of very sensitive and very fragile proteins. Then generally butyl is used. So butyl is a moderate hydrophobic functional group. So general purposes for HIC and has a good balance for your both sensitive proteins and other moderately binding proteins. For strong hydrophobic strength, we have octyl sulfurose, very hydrophobic proteins and detergents. So applications of butyl S, protein purification after salting out, removing protein aggregates or misfolded species, separation of proteins with different hydrophobic surface exposure. So in a group, in a mixture of proteins, you might have some misfolded proteins which you do not need, or they might affect your further interaction studies or whatever you're doing. So if you want a pure fraction of only like properly folded proteins, then also you can do this by removing the protein aggregates or your misfolded species from a mixture of folded and misfolded protein solution. And also it is used as an intermediate step in multi-step purification techniques between ion exchange and gel filtration. So basically, the functional group in butyl S suffrose column is butyl, which provides moderate hydrophobic interactions suitable for general protein purification in IHC. Then we have anion exchange chromatography. So the basic principle for this is the separation is based on charge. Definitely, it's an anionic exchanger, so your proteins are negatively charged. So separation is based on charge interaction between stationary phase and the analyte. In anionic exchange, the stationary phase carries a positive charge which binds to the negatively charged biomolecules like nucleic acid, acidic proteins, and carbohydrates. Elution is achieved by increasing the salt concentration, which displaces the bound anions, or by changing the pH, which weakens the ionic interaction. Then a Dinex Carbo Pack 1 is used here. So this is a strong anion exchange column. The ligand here is quaternary ammonium groups. Function is to bind to negatively charged biomolecules such as RNA, DNA, phosphorylated compounds, oligosaccharides, and acidic proteins. So what is the particular specialty? Is it is widely used in case of carbohydrate and glycoprotein analysis because sugars are weakly acidic and interact well with ion exchange. Others like captophenyl cephirose is used in HIC because it has a hydrophobic phenyl group and it binds to proteins based on hydrophobic patches at high salt and not in ion exchange. Butylex, butylus we just discussed is used in HIC. It is a moderate hydrophobic ligand and its function is same as above, that is weaker hydrophobic interaction than phenyl. Then we have sp sapphirose, which we discussed earlier, is your cation exchange resin. It has a sulfonate group and is a strong negative charged ex- uh, resin. So it binds to positively charged proteins, that is your cations, and not an anion. So this is a comparison between all these. You have your resins, you have your functional groups of your resins, and you have your cations, anions, and hydrophobic protein patches which bind to your cation exchange, anion exchange, and HIC respectively. Then coming to distribution planes. So distribution planes are basically theoretical plates. So in chromatography, the number of distribution planes is given as n or theoretical plates. It represents how many times a solute molecule can equilibrate between the stationary phase and a mobile phase as they pass through the column. That is, each distribution plane is like a mini partition step. More the number of steps, the better the solute molecules separate. So what happens when number of plates or number of planes increase? So more equilibration steps are required, which leads to a sharper partitioning. Solute molecules have more opportunity to distribute according to their affinity. This results in a tighter Gaussian peak. So peak width increases. Uh, Peak width at baseline is inversely proportional to the square root of n, and the resolution also improves. You get narrow peaks, less overlap, and it is easier to distinguish. Basically, you have a better resolution of separation. So in chromatography, a large number of distribution plates indicates better separation between components in mixture. More the number of plates mean the analyte spends more time interacting with the stationary phase, resulting in sharper and narrower peaks. Sharper peaks imply smaller width at the base of the peak. So other cases like the width of the base of the peak increase, this is opposite of what happens if the number of planes are more. Like we discussed, if the number of planes are more, we have a smaller width. Then more plates lead to narrower peaks, not broader ones. And then distance between two peaks decreases. While the peaks might appear closer together, with the increased resolution due to sharp peaks, the actual distance between the peaks do not decrease. The peaks will become just narrow, but the distance between two peaks will not decrease. It's separation that improves, leading to better resolution. And broad peaks would be observed. So, broad peaks are associated with fewer distribution plates and not more. So, it's vice versa. More distribution plate, uh, more distribution plates, smaller width, sharper peaks, less distribution plates, broader peaks, and broader width. Increasing the number of plates will lead to sharper peaks and narrower peaks. So, ion exchange chromatography basic principle is it's a stationary phase negatively charged group with a fixed resin or matrix. Mobile phase is a buffer that carries analytes which has proteins, peptides, amino acids, and binding is your positively charged cation binds to the negatively charged resin. So it's basically inverse or opposite of your anion exchange. So elution is done by increasing the salt concentration. So K plus, Na plus compete and displace your bound protein, and or you can also change your pH similar to that of anion. Your stationary phase matrices. So each type of ion exchanger has a different functional group. sp cephros ff has sulfopropyl group, and strong. It's a strong cationic exchanger. Always a negative charge sulfate regardless of pH, and wide range stability is there. So that's why it's a stronger group, and it is used in separation of proteins with positive charges. Example separates basic proteins like histones, lysozyme, etc. Then CM Cephalose carboxymethyl cephalose, it's a weak cationic exchanger compared to SP. Charge depends on pH, not always fully ionized. That's why it is a weak exchanger. So useful when you want more gentler separation, when you have components which are more sensitive. So that is it. I think this was the last one. So hope I have covered all the topics, all the questions are covered in these topics. If you guys have any doubts, you can ask me right now. You can clarify. Yeah. Does anybody have any doubt or queries or anything else to be discussed? So I will stay online for maybe 5-10 minutes more. If anybody has any questions, you can ask me. The remaining students, if you don't have any questions, can leave the meeting. So this is the end of the session. Thank you, everybody. See you next week. Yeah, ma'am, can you explain? Somebody has asked, can you explain theoretical plates? Okay, so where was it? So yeah, who was that? Yeah, Sai Sampath. Do you want me to explain the entire thing again, or do you have like a particular doubt in it? You can turn on your mic and ask me.
Ma'am, I didn't understand what are these theoretical plates, especially.
Oh, you didn't. You want me to explain it again?
Yes, ma'am.
Okay, just a second. Okay, this one, right?
Yes, ma'am.
Okay, so basically in chromatography, what happens is uh, you have molecules that equilibrate. So that means your your solutes are always in motion and they equilibrate in terms of concentration. So different molecules have different equilibration. So if your number of that equilibration is more, like that is what it means by distribution planes or theoretical planes. So it is between your stationary phase and your mobile phase. So when your mobile phase passes through the column, you have your proteins or biomolecules that are attached to your stationary phase, right? So when your mobile phase passes through the column, your particles should equilibrate between your stationary phase and mobile phase. So equilibrate means nothing like your distribution coefficient or partition coefficient like we discussed. It's similar. It has to partition between your stationary phase and your mobile phase, right? So each distribution plane is like a mini separation step or a mini partition step. So if like there might be number of steps in which it can equilibrate between your stationary phase solvent and your mobile phase solvent. If it equilibrates in like one or two phases, then your number of plates are said to be less. So what happens? You will get a peak right after illusion. So when you get a peak after illusion, because it's just couple of plates, you will have a broad peak like this. But if you have many distributions, your peak will get narrow because like at every step it is getting eluted now. At every step it is being partitioned between your stationary and mobile phase. So your number of plates will increase and your purity or the separation will become more pure and your partition will be more distinct from your previous partition. If you have more number of steps, it will help you separate your peaks with more resolution. That's what it basically means. So in each distribution plane, you what happens is you have a better separation of your solute molecules, which leads to uh, easily distinguish between the separation. You will get more thin peaks or sharper peaks, which will make you identify two different very close by solutes. You can distinguish from each other. It's basically resolution. Did you understand?
Yes, ma'am.
Sure.
Yes, ma'am.
Okay. Okay. Great.
Thank you.
And yeah, yeah, sure. Anything else? Anybody else? Okay, I think if there are no more questions or doubts, I will end the session. Uh, you guys can exit. Thank you. Thank you for joining. Bye, everybody.